Hi, everyone. This is Spencer Childress. I work at Gilead, and I will be presenting safetyGraphics Evolution. Sharing my screen. Community-driven development of innovative tools. So let's get into it. Quick outline. I'll give a little background on the Interactive Safety Graphics group, and then we'll have a little refresher of the Safety Graphics framework, and then we'll get into the new tools and some demos. And finally, we will go over open source best practices. So the ISG is an open source collaboration between clinicians, statisticians, medical monitors, and technical contributors like R programmers, statisticians who like to code, data scientists, and open source enthusiasts who want to contribute. And the focus is on safety evaluation from a more interactive, innovative mindset that kind of eschews the static, long summary tables for more interactive, drill down safety visualizations. So a little refresher on the Safety Graphics framework. It's an R-Shiny framework. It's very modular and it's completely open source, as is all of the tools you'll be seeing today. And the nice thing about Safety Graphics is everything's metadata driven. And what does that mean? So the data mappings from your input data to the data expected by the modules is captured in mapping metadata. There's a UI for that within the app. The modules themselves are also metadata driven. So really, which data sets do they need? Which domains do they rely on? And which columns are used within each module? This is all captured in metadata. And finally, each module specification includes a number of additional information like references and associated R packages to find the code. And finally, a few design principles of the Safety Graphics framework: reproducible code. So each module you see is exportable to run locally within your R session. There's data transparency, so you can see the data at the second tab in the application and understand what's going into what you're seeing in each module. And then finally, everything's exportable. The mappings, the module specifications, the code, and the data. Careful with that, but everything's available for export. So let's get into the demos, the fun part. The first one we'll look at is the safety profile. And here, let me just refresh this one real quick. So the safety profile is a participant level application. And within the application, you have submodules for various data domains. And the reason you would want to look at something like this is to drill down from kind of a high level clinical trial analysis population view and down to a single participant and see what's going on with them. So going back a step, this is incorporating multiple data domains, and really you can adapt any data domain with the appropriate view of the data. So for instance, this plot you see on the right side is an event timeline, and you're looking at the adverse events and the concomitant medications for this participant. You also see we have a couple other tabs, an overview tab of the data for this participant, as well as a measures over time tab. And so let's go take a look. So default landing page for the Safety Graphics app. On the second tab, you can see our mappings, input data domains, and the mappings of their columns to the charts over here. We can define a custom analysis population here. This is all kind of pre-existing functionality. And then now we have this new profile app. And you can see up in the top right, we have a little pill indicating which ID we're looking at. You can see a drop down right here where we can flip between IDs, and you get an update on demographic information about this patient. You also get their data subset to their records, and then you get modules for each input data domain. And so the event timeline, which we've already talked about, we're looking at the adverse events for the subject, and the measures over time is actually a view of the lab data, which we'll come to last. There's a little bug between this module and some of the charts that that safetyGraphics kind of ships with by default, and we're working on that integration. So I want to highlight 5 of these default charts in safetyGraphics. Start with the adverse event timelines. And right here, you see all of the participants in this data set and their adverse events. So we can click on one, and you'll see, okay, this participant has 4 adverse events. And you see our pill updated to this ID, and we can quickly navigate back to the profile and see those 4 adverse events, along with the other data associated with this participant. So in addition to that chart, the Outlier Explorer gives us the same capability. So once it loads, we can click on one line, and within this module, you also get a little patient profile below of all of their labs. You see we've also updated the ID up here. Similarly, the shift plot change from baseline in a single measure gives you the same functionality. You can brush a point. You can see our participant ID right here, updated up here. Delta plot gives you something similar. This chart is looking at the change over time between 2 measures. So right now we're looking at this measure against this measure. We can click on one of these points, update our selected ID. And finally, the hepatic safety explorer has the same integration. You can click on an ID and see that 1239 populated up here, populated in the profile. So this participant that we just clicked on, they have, it looks like 9 or 10 adverse events. And now we can take a look at this this module specific to the lab data, which basically gives us their lab results over time with a little expansion to look at the individual data points. So that is the patient profile. Really nice, powerful integration between the existing safety charts and this new module that lets you drill down into a patient's data. The next one we'll look at is the Nephrotoxicity Explorer. Nephrotoxicity is the decline in kidney function due to toxic substances like those introduced in a clinical trial. This one's pretty similar to the Hepatotoxicity Explorer that we just looked at. We're just looking at different analytes, including creatinine, glomerular filtration rate, cystatin C, things that define kidney function and give you some insight into how this drug is reflected in kidney function. And this is an application of medical guidelines to lab results for a framework of nephrotoxicity oversight. So let's go take a look at this one. You'll see we have this color palette of yellow, white, yellow, orange, red, kind of indicating the severity of the level of kidney function. And we're actually looking at the fold increase from baseline plotted against the absolute change from baseline. You'll see if you meet either of these criteria, you fall in the red. So you don't have to meet each one. I think either one kind of tags you. And if we click a point, we can actually see these individual analytes over time, or individual metrics over time. So this first one is percent change from baseline. You can see the ID I clicked on has a 200+% change from baseline. Let's see, 3.27, 2.224. These don't, well, yeah, there we go. So that point corresponds to that point. We have the raw change right here, and then we have some additional lab results that give us information like the GFR, and other lab tests, as well as vital signs. So again, this is another multi-data domain application that we're still working on integration with the safetyGraphics system, but expect this one soon. Next, we have the qtexplorer. It's an electrocardiography data explorer. And we're looking at electrical activity of the heart. Change in electrical activity can be expected with certain therapies, and this tool is oriented to that data, to that ECG data. And again, we're applying kind of medical guidelines developed by subject matter experts to ECG data to provide a framework for arrhythmic risk. Let's go take a look at this one. You'll see this one is integrated within the safetyGraphics framework. You'll see we're looking at demographic data and ECG data. Let me make sure all of the, yeah, we're good here. All of the mappings are good. So we have our participant selector. We can create a little analysis population, and you'll see a number of these are charts we've already looked at. The box plots, let me rephrase that, charts that have existed previously. Box plot, histogram, shift plot, paneled outlier explorer, delta plot, and spaghetti plot. But at the top, the two new ones, we have the QT central tendency plot. And the first, so this view is basically just giving you your aggregated ECG result over time by treatment with some built-in thresholding. So we have 450, 480, 500. And the reason these thresholds were chosen is surely described in the literature. We also have this scatter plot view of individual patient data. You can see it's stratified by treatment. And right now we're looking at this QT interval. So this is the kind of distance between the Q wave and the T wave and the ECG. And we're looking at change from baseline plotted against the baseline value. And you can see all of these kind of thresholds, and we can add and remove them. But these thresholds kind of give insight into the ECG result. I cannot go into any more detail than that. But we have this baseline against change from baseline or observed, and we also have observations over time. Oh, this one's cool. So this one lets you see all the points move over time. A little crazy. And then we have just some information, some data quality information, like how many data points do we have at each visit. So that's the QT chart, another new data domain. Now we have the Volcano plot next. This is built on an existing data domain, which is adverse events, and this is going to give. This kind of replaces all of those long adverse event summary tables by body system or system organ class and preferred term, and it kind of captures all of that information and the relative incidence between treatment groups in these nice Volcano plots. yeah, so it's very succinct, if you will, compared to how things have been done in the past. Let's go take a look at this one. So again, you'll see that this one is not in the safetyGraphics framework. We are working on an integration, but as you can imagine, it's built on adverse events, so we can map it into the framework. So you have two statistics that you can look at: risk ratio and risk difference, which are plotted on the x-axis, and then on the vertical, you have the log p-value, significance of the incidence rate between the two treatment groups. So in this left pane, we have placebo, and in the right pane, we have one of the treatment groups, and these are being compared against the third treatment group. So if we go look at, say, this value, which is going crazy, let's look at that one. There we go. So you can see we're comparing the reference group, which is the low dose, against placebo and against the high dose. You can see that, okay, over here, it's, up in this quadrant, it's significant. We can also look at the data that goes into this statistic. So in addition to risk ratio, we can look at risk difference. And then we can also look at, instead of by system organ class, system organ class, we can look at by preferred term. And a preferred term is basically a subset of a more granular subset of body system. You can see, we have a number of different comparisons, as well as this little blue guy. Not sure what that means. But anyway, it's a nice tool to look at, dig into adverse event data. And then finally, we have this benefit risk forest plot, which is a benefit risk assessment tool of let's say, a novel treatment, where you can kind of quickly visualize one or more benefits and one or more risks, and kind of come to a high-level decision about the favorability of the therapy. Yeah, and again, it informs pre and post-market regulatory decisions. And a lot like the previous chart, you're looking at summary statistics rather than participant level data. So we're actually, working with an aggregated data set instead of a participant level data set like adverse events or demographics. So yeah, let's take a look real quick. And this one also is integrated. However, I don't believe that the aggregation has been developed within the module. You see, you land on this kind of view of a forest plot. You have the control on one side, the active treatment on the other side, and you can kind of see, okay, these benefits are obviously, they favor active, and these risks favor control. We can look at subgroups like the dose. Okay, so this, this dose is more risky than the other doses, and also more beneficial. We also stratify by other participant level stratification factors like sex or race. But yeah, I think that about covers this one. It's a nice concise view of benefits and risks. So that concludes the demos. Let's just recap some open source best practices. So inclusion, make people feel wanted, and kind of adapt tasks and work to people's strengths. And then kind of check in with everybody regularly. Look at, kind of it's good to see each other and get away from the GitHub notifications or the emails, and actually actually talk. So have regular working hours helps. And in the safety graphics group, we offer these office hours every other Friday, where people can tune in and we can debug issues with their safety apps. I think it's a valuable resource for teams. And then industry outreach. The people who work on this project are from a number of different companies, and every company is kind of doing things a little bit differently. So just having a broad industry, cross-industry group just helps kind of helps have more ideas and bring more input into decisions. And and kind of really give contributors ownership of these projects, and and I think this empowers people to help out and to be accountable to the project, and just really own it. And then finally, reproducible examples. When you finish up a pull request, just make sure your code's rerunnable so that others can understand what's going on and help you out. So that concludes my talk. Please do join the team. You can see a list of GitHub avatars up here in the top right. We have 60 on GitHub that have contributed to this initiative, and here's just a few of the names right here. And yeah, please join the team, and thank you for attending. Thank you for attending this talk. Let me stop sharing and stop the recording.